forgot that I double booked a webinar today. So before I could drive back to the other side of the state, I am borrowing one of their corner offices to give this webinar. Yeah, so my dissertation research is in created wetlands and wetland mitigation, and sirens are not really part of that arena. They are, I think, as my advisor called them, a distraction. But as I share the story with you and I share this work, I hope you'll agree that it's a pretty worthy distraction because it is a really cool story. And let's get started. Hopefully, there we go. So just to start out, for those who maybe aren't familiar here in Michigan, sirens were first discovered back in 1958. The DNR was going to Saddle Lake in Van Buren County, and they were applying rotenone, which is a pesticide, to a carp population. And they found a couple of these dead creatures washed up on shore. The residents were concerned. They called in, and one of the DNR's herpetologists came out there and identified them as lesser sirens. So that was the first time they were ever documented in Michigan. That was 1958. And then 5 years later, they found another dead one, again, from a rotenone application at a nearby lake. And since then, there hasn't been any documented records of sirens occurring in Michigan. And so we've kind of been in this state of limbo of, are they here? Are they not? Do they deserve protections? Don't they? So first, what is a siren? When I try to talk about what sirens are to people, I think everyone's frame of reference tends to be a little bit off. If I say siren, a lot of folks might think of maybe the sirens from Greek myth, and they're not sirens, or maybe something a little bit more modern, like, you know, an alarm siren, obviously not even close. So maybe I might describe them as an animal, or like these aquatic salamanders with these external gills, and they'll be like, oh yeah, my daughter has a siren in her aquarium in her tank. And that's not a siren. Axolotls and mud puppies are entirely different clade. And then maybe I'll describe it to local sportsmen or outdoorsmen. You know, I'm trying to figure out where these things might be found, if anybody's seen them. And they're like, oh yeah, I've seen a siren. It was going after my fishing bait the other day. We saw it swimming in between the lily pads. And usually those are northern water snakes. So what sirens actually are salamanders. They are members of the family Sirenidae. They are these fully aquatic salamanders. They're very eel-like. They're very eel-shaped. They even have that long, flattened, paddle-shaped tail, so they can move around really quickly through the water. They do grow quite large. They can reach up to 2 feet or more. And some members of the siren family can get up to over 3 feet. And one of the things that distinguishes them very clearly from mud puppies and axolotls is that they don't have rear limbs. They have 2 front little legs and nothing else. They don't even have a pelvic girdle, so they don't even have those vestigial traits that would indicate that they once had hind limbs. Sirens are part of that group of salamanders that we would call neotenic, and that just means that our mature sexually reproducing adults have very similar characteristics as juveniles. Normally, with a lot of the salamanders we're familiar with in Michigan, they're aquatic, they have those external gills, but then they metamorphose and they become terrestrial and they lose those gills. Sirens retain them for their entire lives. A little bit of the natural history of the family. So there's 2 genera within the family, comprising about 8 different species. So there's the siren genus. This includes the greater siren, the eastern and western lesser sirens, the Rio Grande lesser siren, the reticulated siren, which was just described in 2018, and even more recently, the seepage siren, which was described in 2023. And if you look at all these maps of their currently known ranges, which are based off of iNaturalist observations, almost all of these siren species are concentrated in the southeastern United States, or in the case of western lesser sirens and Rio Grande sirens, they're here in sort of the deep south, Mississippi Delta region. They are a very basal salamander clade. Some might call them primitive. I'll say basal, meaning they split off from the rest of the salamander groups about 165 million years ago. So most people, if they're thinking of gilled salamanders, neotenic salamanders that we have in Michigan, they might think of mud puppies. And sirens diverged all the way back here, and all the way up here, Proteidae are mud puppies. So these are They have similar traits, but they're not especially closely related. Someone once called or described these salamanders to me as the anti-salamander. Most salamanders that we're familiar with do pretty well in the cold. A lot of times they're out breeding while there's still snow on the ground. And they need these really sort of pristine wetland conditions or high oxygen conditions to survive. But sirens are really adapted for these hot, arid, or seasonally dry conditions that you would find sort of in the deep south. So they can estivate in these mucus cocoons underground. So if they're living in a shallow wetland and it dries out, rather than having to go search for a new one, they can just build this mucus cocoon, dig underground, and they can sit there for 6 months or more, depending on their nutrient reserves. They're actually very cold intolerant. So we've had some polar vortexes come down, and we've had some mass mortalities of sirens in southern Indiana, where we have some populations down there. So they do not do very well with freezing temperatures. Unlike a lot of other salamander species, they don't necessarily need these pristine high oxygen conditions. They actually prefer waters with these deep organic sediments, the kind of water where the water is maybe 6-7 inches deep, but if you actually stood in it, it would go up over your head because there's all of that muck and that false bottom. So they're very tolerant of eutrophic, low oxygen, high nutrient conditions, but they're still probably very susceptible to human-introduced toxicants. So it was back in 2018. I was walking down my dock and I noticed a little string in the water tied to my dock. And I remembered that earlier that year in the spring, I set out a minnow trap. I was just trying to collect some fishing bait and I'd forgotten about it. It had been in there that whole time. So in August, I pulled up this trap and I found one of these dead salamanders in it. And I didn't really know what to do with it. I didn't immediately recognize what it was. I wasn't expecting to find anything. And I said, that's really too bad. That sucks. And I just kind of tossed it back in the water. And it wasn't until I got home and started thinking about that and what it probably was that I realized that that was a lesser siren. So I called the DNR. I tried to report it to the Michigan Herp Atlas, but I didn't have a photo of it. I didn't have a body. And they sort of said, let us know when you have a photo or a voucher or something like that. So I began doing some intentional trapping on that lake, just unbaited minnow traps. We put out 10 to 20 of them, and I would check those every other day at least, usually every day. So every day getting home from work and then kayaking around a 70-acre lake and pulling up these traps to see what we could find. I didn't find anything that fall, but then in 2019, we photographed this guy, which was the first living lesser siren that's ever been found in Michigan or even in the Great Lakes Basin. So that was spring of 2019. Since then, we've been doing some long-term surveys at that lake. So we've continued doing that same sort of trapping with these unbaited minnow traps. We set them in conditions where it's just, the water is just deep enough to cover the entrance holes all the way out to 1.5 meters deep. So we've had 20 of them sitting around this lake, and we're checking them every other day. We did that year round from 2019 to 2020, and then we switched to only checking those traps and deploying them March through May from 2021 to 2023. And so whenever we found some of these sirens, we would anesthetize them in 1 gram liter buffered MS-222. They're a very slippery species, so it's hard to hang on to them. It's hard to work with them without putting them under. We would take some morphometrics, some body measurements. We put a microchip in there in the tail muscle. We'd take a toe clip as a DNA voucher, and we'd let the sirens recover fully in nice, cool, aerated water in a bucket, and then we returned all of them awake and, other than the toe clip, mostly unharmed to their capture location. And so all of them were released relatively healthy. So after 4 years of doing those surveys, so this is more than 1,200 person hours. This is more than 5,000 trap checks. We found 14 adults, 1 juvenile, and one of those adults we recaptured. So a total of really 15 captures. And the reason we stopped doing year-round surveys and I didn't have to continue chipping through the ice like you see here to pull up traps is because we were finding that we only found these animals entering traps through this March through May time period. Anytime outside of that, at least for us, we were not able to find them. So we did confirm, especially with that juvenile, that we have at least one breeding population of sirens in Michigan. And as a result of us finding a breeding population of living sirens, they were uplisted to state endangered in 2023. Now, this brought on a lot of questions. So if you look at this map here, you have their initial observation, that 1958 observation in the Black River watershed. Again, over in the Kalamazoo River watershed, and then where we currently know of them in the Paw Paw watershed. So these are not hydrologically connected systems. These are separate watersheds, and there's a lot of farmland and intensive agriculture and roads between these. So we're kind of at a loss of how are they getting around the landscape? They must be in more locations than just that. So the first question was, where are there other populations of sirens in Michigan? We're also not really sure if sirens are even native to Michigan. They probably are. We're probably at that northern extent of their range, but they could have also been introduced by humans, either as a released pet or, fishing bait that was brought up, from maybe Indiana or someplace, further in the deep South. We don't really know that right now. And the other one is this actually the Western lesser siren, or is this an undescribed or new species or subspecies? So right now, with Sean Graham's work and Matt Fedler's work down in Florida, they've been finding a lot more species of sirens than we previously thought existed. So with this one being up here in Michigan, very different environmental conditions, a lot of geographic and reproductive isolation from other populations. Just kind of wondering if maybe this is something different altogether. Well, we set about trying to answer that first question, especially trying to locate more of these animals and more of these populations so we can study them and get a better idea of what type of protections they might need and what type of habitats maybe they associate with here in Michigan. And one of the tools we decided to use was environmental DNA. So you can think of environmental DNA as like a forensic technique. You are sampling the environment, looking for the DNA of the species that you're interested in. And so in our case, we worked with a commercial lab, Jonah Ventures, and they developed this new eDNA assay for us that is targeted specifically for Michigan's population of sirens. We sent them tissue samples that we had collected, those little toe clips, and they developed this assay. We tested it with positive controls, so lake water where we knew the sirens were at. And we also tested it with negative controls, so tap water, maybe a garden pond where sirens had never been introduced. And we were able to verify that the assay was working, and then we went ahead and deployed that. So the idea behind this was, let's go through these different watersheds, and let's trace back all these little hydrological connections, what we call warm water transitional streams. These are like those creeks you see on the side of the road, those farm and ag ditches that connect all of these lakes and all of these little streams to the larger river. And the idea is that if you sample periodically along it, especially at major intersections, if you have a positive detection on one branch but not on another, it can be assumed that maybe if you have a positive here, you might have sirens residing in a habitat that's upstream. And the idea is that we're sampling the water, then all of that DNA must have come from somewhere. It must have come from upstream. Okay. So we tried this. We looked at 103 different locations across 5 different watersheds. So we have our historic locations, highlighted right here, as well as our known location. We've got an unconfirmed report up in the Muskegon Lake watershed, and we went ahead and sampled all of these different points. We sent in those water samples into the lab to be analyzed, and they did a process called quantitative PCR, which is just trying to figure out how many copies of DNA per milliliter are present in that sample. And so they would run this qPCR 3 times. And if we found DNA in at least 2 out of those 3 replicates, we considered that a positive detection. So when we put this on a map, all of these different pins represent points where we sampled for the DNA of these different sirens. And then we made a heat visualization of all the positive detections, right? So all these empty pins are negative detections. The positive detections are filled in. Any of these blue diamonds or orange stars are considered positive detections. And we got this really, dense heat map right around where the historic observations were, and that was kind of expected. And then we got this other unexpected sort of cluster over here in Southwest Berry County of positive detections that we wanted to have a look at. A couple other findings that we got from this eDNA study. When we were pushing water through those filters using that little syringe, we push that water through until it clogs. And we thought, the more water we push through these filters, the more DNA or estimated copies of DNA that we're going to find. And what we actually found is that for samples where we pushed more water volume through those filters, we actually ended up with less DNA per milliliter. And we're kind of wondering why that might be. One of the things that we kept track of at each one of these points where we sampled the water, we also kept track of the sediment type. So if we were pulling water from a creek, we weren't getting in the creek, but we were at least noting what type of sediment was in the bottom of the creek. And so we classified as clay, muck, sand silt, or silt. And we found that depending on the sediment type, it influences, for one, how much water you can filter. If there's a clay sediment or a muck sediment, those clog up those filters really quickly. We can't filter as much water. Whereas coarser sediments, we could filter a lot more. But also, it had an effect on the numbers of copies per milliliter that we were able to find. So clay and muck substrates were much better at yielding DNA than sand or silt. And the reason I'm pointing this out, it seems maybe a little bit boring, but eDNA has been kind of used as this tool. Folks are trying to use it to detect species, but not only to detect them, but maybe to estimate maybe how many there are or what their abundance is. But what we're finding is that the amount of DNA you find in a sample is really strongly influenced by environmental conditions. Something as simple as what type of sediment you have is going to influence how many copies of DNA you're finding from that sediment. So even though we might find more copies of DNA at one site, that doesn't necessarily mean that there are more sirens in that area. Well, we had these positive detections, right? Southwest Berry County, no previous records of them. And so we decided to say, hey, if we've got positive eDNA detections, let's go out to some of those spots where we got those positive detections. We'll look upstream, we'll find some likely habitats, and then we will start trapping just like we did at their known location. And so this spring, 2024, we deployed 30 pairs, so 60 different traps. We used box and cylindrical minnow traps, unbaited. We had those deployed out there March through May. I don't know why it says April. March through May 2024. And we were checking those every other day. And we were really optimistic that these eDNA detections would lead to some sort of actual visual observation, that we'd be able to trap sirens at these locations and maybe find some new established populations. And here are kind of the search results. So again, looking at that known location where we had been surveying for years, over 4 years, we got those 15 captures. And if you look at only the March through May time period, it was 1,440 trap checks. So that's about a 1% success rate. If you go out through that March through May season, you can expect that, pulling in a trap, you get maybe 1 out of every 100 will have a siren in it. So then we went out to those eDNA positive locations. Just in this 1 year, we went through that same time period, and we did 1,874 trap checks, right? And 0% success. We didn't find any. And so we would expect that if they were there, maybe, we would capture them at a similar rate that we do at this other location, but we weren't able to find any. And what you see here on the left is a species accumulation curve. And so we have the trap checks on the x-axis, and we have the species richness, or the cumulative number of species we found, on the y. And so we just kept trapping the mess out of these different locations. And eventually what happens is it levels off. That means you found all of the species that are available there to find. And in almost all these 6 sites, we got to that point where it just kind of leveled off and we weren't finding anything new. So it was very unlikely that we were missing anything. And then we did some richness estimations as well, sort of statistical techniques to try to determine whether or not there are species that maybe are there in the community that you just didn't detect. And the estimated richness was coming back as very similar to our observed richness. So we're thinking probably there weren't sirens in these locations. The question was eDNA an effective tool for us? It promises to be a pretty effective tool. It's been used for other studies, finding aquatic salamanders, amphibians, fish, including sirens in the Southwest. And it was an exciting thing to try. What we found, for one, is that our positive detections were usually downstream of some suitable habitat. So these are normally lakes that are deep enough where sirens are not going to be trapped in freezing conditions. They can get down deep where there's still a little bit of thermal energy, there's a little bit of warmth that they can overwinter in. So these are eutrophic sort of kettle lakes that are formed from glacial activity in Michigan. We also found that eDNA is really powerfully influenced by environmental factors. And in our case, at least, eDNA did not inform successful manual surveys. What we can say is probably sirens were likely absent at these locations. Now, we can't prove a negative. Michigan, we went 56 years without finding sirens, even though there had probably been, some surveys for them. The DNR had probably been looking for them. So we can't prove a negative. We can't prove that they weren't there, but we can say, based on the trapping effort, that they were likely absent. And so as much as I think eDNA could be an emerging and useful tool, right now there really is no substitute for direct firsthand observation. So we've got some known unknowns. We still have these outstanding questions. Where are other siren populations in Michigan? Obviously, these surveys are labor intensive. If we're going out every other day, checking a series of minnow traps and documenting what we're finding, and we're only catching one siren out of every 100 trap checks, that's a lot of labor to go through for one species. These habitats have false bottoms. They're very weedy, especially during the summer. They can be quite deep, especially here in Michigan, at least, so they can escape those colder temperatures. And so backpack electrofishing, even from a boat fishing rig, doesn't work too great up here. It might work a little bit better in the south, where you have shallower systems that you can work in, but it doesn't work too great up here. Bob Broadman, who was trapping lesser sirens in Indiana, also did a study on whether or not baiting these traps increases the capture success, and it doesn't. It actually ends up attracting some predators. So you don't get more sirens, but you do get a lot more crayfish. And I don't know if you can see in this little short clip here, I'll rerun it. This is one of the sirens we captured. And we were checking these every day, and it had already had a pretty big chunk of its tail taken out, as you can see it coming back around there. And that's usually from crayfish or other sort of large macroinvertebrate predators that also get into these traps with these animals. So if you're going to survey for these, recommend unbaited minnow traps. What I think is really cool about sirens is this is a great opportunity for citizen science and collaborative surveying. And normally in the herp world, especially when we find these rare species, there's a need for a little bit of secrecy and guarding where the locations are. In this case, it might not be so necessary. There are least concerned species federally. And not only that, but when you're worrying about people, who might come out to collect them or something, they would have to trap for months on end with hundreds of traps to just get a small handful of them. And even at those few individuals are probably not going to affect the population size. So this is one of those rare species where, or at least rare in Michigan, where you could probably incorporate some citizen science volunteers, distribute some unbaited minnow traps for surveying and likely habitats, and you're unlikely to have any significant population impacts from people over-collecting or over-sampling them. We also don't know about their origin still. This is still an open question, right? So we don't know if they're native or introduced. We don't know how they got into Michigan. Again, Western lesser sirens especially tend to cluster around the Mississippi watershed, all the way up into the Wabash watershed, but that's a separate watershed from the Great Lakes Basin. There used to be this really expansive marsh system, the Kankakee Marsh, that is also known as the Everglades of the North. It is now mostly farmland in northern Indiana, but historically this would have been a very expansive wetland system where conceivably sirens could have moved through. There's also a great PBS documentary you can look up. I think it's free to watch, called The Everglades of the North, if you want to learn a little bit more about that wetland system. But they could have come up through there into Southwest Michigan. Again, they could have been released pets. They could have been escaped fishing bait. And in the deep south, where these are a little more common, it's not unusual to find sirens being sold as pets or as fishing bait. So to answer these kind of questions, we really need some individuals who might be interested in getting into population genetics work. If these are sirens that are coming up along those natural corridors or came up along those natural corridors from Indiana, we would expect that they'd be much more closely related to those from Indiana than those from, the deep south, where they're much more common. We did send in a couple of our samples to Matt Fedler, who's described the seepage siren recently and has been working on sort of a range-wide phylogeny of sirens. So he looked at a couple different RNA and mitochondrial DNA segments. And again, these are toe clips. There's not a lot of DNA yield from them. We only sent in a couple samples, but they kind of placed right in there with all the other SI natingi, other Western lesser siren clades. So we're thinking probably these are sitting right there with other Western lesser sirens. They're probably that same species, maybe not a new species, maybe not a new subspecies, but we still at this point probably need a little bit more work into their phylogenetics. Because again, this is, new siren species are being discovered and described all the time. We have a ton of geographic isolation with this population all the way up in here. Its nearest population, 100 miles south, and then further on into the deep south. And so right now that phylogeny I was just kind of giving you a glimpse of is based on 2 samples with very limited DNA yield. So looking at some conservation implications for these guys, which I know a lot of folks are interested in. And the first one we might talk about is climate change. Climate change has some really substantial implications for this global amphibian decline that we're seeing. It's impacting a lot of our cooler, montane, temperate species. It's facilitating the spread of chytrid fungus and ranaviruses and these sort of things that are coming in and decimating populations. But sirens are one of a few species, at least the sirens in Michigan, that are likely to be climate winners. And a lot of times when we talk about what the climate looks like in the future, we talk about winners and losers, so species that are going to benefit, species that are not. But if you look at where sirens, especially Western lesser sirens, are clustered, they're clustered right around that deep south Mississippi Delta area and following up in the Mississippi watershed. And then you compare that to, Catherine Hayhoe's work looking at Michigan's different possible futures for our climate. It's looking like we're heading towards their preferred climate type. So the good news is that climate is probably not going to be a substantial impact on lesser sirens. They're pretty well adapted to heat and to temporary wetlands and to long dry periods. Toxicants are another issue entirely, especially pesticides like rotenone. They obviously cause direct mortality. These were how our first 2 populations were found. So, I guess if you really wanted to find sirens, you could dump that in there and you'd find that and everything else floating up to the top. Herbicides, so things like diquat, triclopyr, endothol that a lot of us land managers rely on in combating aquatic vegetation. Again, these guys really love aquatic vegetation. That is their preferred cover. That is how they take refuge from predators, especially in systems up here in Michigan where there are year-round predators. We don't know the direct effects. A lot of the maybe clawed frog larva or on zebrafish. But insecticides, things like atrazine that are used a lot in agricultural communities, are powerful amphibian hormone disruptors. So we look at Tyrone Hayes' work out of UC Berkeley, and looking at the effects of atrazine on African clawed frogs. You can see here kind of an image of what happens to them when they're exposed to atrazine. So just kind of an idea of what it is. This is a male frog that now has ovaries and testes, and another set of ovaries and another set of testes. So, and over here you have an image of a control male frog, and then another one that's been exposed to atrazine. And the frogs that you see here mating are actually genetically male frogs. They've been exposed to atrazine and feminized, and they are successfully reproducing to male frogs, and they're producing fertile offspring that has never that have never themselves been exposed to atrazine, but are suffering from those effects. So we can imagine that similar effects could be impacting sirens. Now, I do want to really emphasize the toxicant thing. So we discovered this population, this breeding population of lesser sirens. They've been missing for so many years. It might be something new, something novel. And it wasn't even a year after we discovered them when many of the lake residents wanted to apply diquat, which is a non-selective herbicide. It's used for kind of temporary weed control. They wanted to apply this herbicide to the entire lake to try to help with weed management. They were getting tired of, raking their beaches. It's a little bit of a eutrophic lake. It's got a lot of nutrients. And that permit was eventually denied, but it was denied solely because sirens were present there. So that could be a potential impact and something to consider. Predation is another one. So in the deep south, where these guys are relatively common, they can be the top predators in these shallow temporary wetlands, these drainage ditches, agricultural ditches, things that'll dry out seasonally. They'll aestivate down in the mud. Fish can't really survive in those, and you can get really high densities of sirens there, as many as 1,000 sirens within just a little quarter, acre wetland. But when you get up to Michigan, those shallow wetlands don't work anymore because then they're being exposed to freezing temperatures, and even burrowing down doesn't get them below the frost line. And so they kind of occupy these deeper kettle lakes, or at least we think so. And so that allows them to overwinter here, but it also exposes them to a lot of different predators and predation pressures that they might not experience elsewhere. And so this is likely a contributing factor for why we don't find them in the same population densities and the same high numbers that we might find in the Deep South. Finally, on to management actions, just kind of to close this out. I am desperately in need of folks to survey for these guys. Some unbaited minnow traps, the kind that you get at Bass Pro, just deploying them in some of these shallow, weedy areas, especially in Southwest Michigan. If you have the time or resources or know folks who'd be willing to do that, that would be great for helping us find them. The eDNA didn't work out too well for us. But yeah, as far as I know, I think I'm the only person working with this population here in Michigan, and it would be nice to know that there are some other populations out there. For land managers, just considering whether or not vegetation management actions might cause more harm than good. A lot of times we see these invasive, like curly leaf pondweed or milfoil, and we want to go in there with mechanical management and get those out of there and establish native vegetation, but these guys can also be wrapped up in that. So if we're concerned about conserving these guys, then maybe we need to think, do we really need vegetation management here? Is it going to be for the broader benefit of the species that live there, or it's maybe better to sort of leave things alone, at least for that location? And then avoiding chemical treatments, especially near water bodies. So with that, a couple of people to thank. Tiffany Schriever at Western Michigan University is my advisor, and she has been not only very supportive and given great advice throughout this project, she's also been very patient in how much it's distracted me from my dissertation. Savannah here, who is highlighted in green, is an undergrad who worked with me in collecting those eDNA samples, and she did all of those 1900 trap checks this spring out in all weather conditions. So just absolutely a beast with collecting data. Morgan and Nicole both helped with some field work. some limited surveys that we did, and I also received a lot of funding from the Pierce Cedar Creek Institute to do that eDNA work and the follow-up annual surveys. So I think that's it for me. And at this time, I guess I can open it up to any questions.
Well, great presentation, Adam. Thank you for that. We do have a handful of questions, if you don't mind. I will just knock them out as they come in. So if you attendees have any other questions, be sure to type them in as we're answering the questions that have already been submitted. The first question is, do you think that the sirens might be attracted to a submerged light?
You know, there's been some studies recently about light baited traps. They could. We do have some captive sirens. I have some captive sirens that are from further down south, and what I can say is that their vision is really poor. They find their way mostly around with a Jacobson organ, so they're detecting chemical signatures in the water. They pretty much can't see anything right in front of them. They can kind of just tell if it's night or day. So maybe those lights would attract them. I don't know. I don't know that anyone has done any light baited studies for lesser sirens, but if it works with sirens like it does with other amphibians, it would be a really cool thing to try.
Mhm. All right. So do you know if anyone has ever caught a siren on a baited fishing hook?
I don't know of anybody. I'm sure it could have happened. I'm sure that it's conceivable somebody caught one out there and thought that it was, a mud puppy, or I think a lot of folks think like snakehead whenever they see something that they don't recognize. Um, it could have happened. They will eat earthworms and that sort of thing. It hasn't happened in all my years fishing that lake where we do know that they're at. So, yeah.
Okay. The juvenile siren that you found, is that the youngest one you've ever seen?
Yeah. Yeah. So, sirens, they start out, these really cute little guys, maybe, a couple inches long. And within the first year, they can get to full adult size, so, 15 inches or more. So just finding one that was, I think that guy was about 6 or 7 inches long. That was probably the smallest one that I saw. And we knew that he was probably in his first year or two of development.
Okay. You mentioned predation. I mean, maybe we don't know this, but what are the predators? Should bass fishermen be checking stomachs for evidence of sirens?
I'm all right with that. If you're going to pull out a bass or one of these top predators and you want to report fish gut content, that'd be awesome. I would assume that those are going to be top predators. Sirens, there's a reason that a lot of those rubber lures around out there resemble sirens. They are absolutely a great snack for fish. That is how they play part of their role in the ecosystem. They're invertebrate predators and they're prey themselves. So yeah, bass, bowfin, catfish, pike, all of those things I could see consuming sirens.
Okay. Are you familiar with Michigan Natural Features Inventory Rare Species Review?
I am,
Okay,
yes.
so due to your work, will Western Leicester sirens show up on those MNFI reports near the lake you found them, or how is that factoring into MNFI reporting?
Yeah, so all of our observations at that lake have been reported to MNFI, and back when that permit was applied for the herbicides, they had not yet been uplisted to endangered. They were in that process, but now that they are, they should be part of that surveying. The problem is, of course, there's only one lake where we know of them at, and so we need some other confirmed observations and more data coming into MNFI for that to really be useful.
Okay, this is not totally, I mean, it is a question, but um, somebody is volunteering the information that there is a conservation stewards program through MSU Extension taking place in Berrien County. Those students need to do a capstone project. Might you be willing to work with a student on doing some surveying in the area?
Absolutely. I am willing to help set up anyone with those efforts. Again, I can't be out there all the time, but if anyone wants to get started surveying, if there's any researchers out there who want to even work with our known population, I'm happy to help facilitate that.
Okay. And so how do we contact you?
You can contact me through my Western email, so adam.r.austin@wmich.edu, or my Rattlesnake Conservancy email, so austin@savethebuzztails.org. You can also
Okay.
Google me, my contact info.
All right. Do you think, as folks are filing out, that you could perhaps type those email addresses in the chat directed
On it
to
right
everyone?
now.
Okay, got it. Thank you. All right, let me see if there are any more questions. Okay, no more questions at this time. So here is a final opportunity to submit any questions you might have. Otherwise, feel free to leave the webinar. Thank you, Adam, so much for discussing lesser sirens with us. It's really cool work. How cool for you to find them after so many years. We really appreciate your time.
All right. Thank you so much, Nicole.
All right, thanks everyone. Have a good rest of your day.